Deep inside a eukaryotic cell, the cytoplasm is a dense, chaotic environment. Floating within this fluid is a long, twisting strand of messenger RNA. This mRNA strand is a highly processed copy of the cell's genetic code. It arrives fresh from the nucleus, capped at one end with a protective methyl group, and trailed by a long poly-A tail at the other to keep it stable in this volatile environment. But here lies a mechanical problem. A sequence of nucleotides carries vital instructions, but data is entirely passive. A sequence of letters cannot physically contract a muscle, build a cell wall, or digest a piece of food. For life to function, the cell needs a physical mechanism to decode this one-dimensional string of information and translate it into three-dimensional, working machinery. The building blocks for that machinery are the 20 distinct amino acids scattered throughout the cytoplasm. To be useful, they have to be transported to the assembly line by specialized vehicles known as transfer RNAs or tRNAs. Getting them onto those vehicles requires brute force. An enzyme called aminoacyl tRNA synthetase grabs a specific amino acid and violently snaps it onto its corresponding tRNA. This loading process burns cellular energy in the form of ATP. That energy expenditure acts as a quality control mechanism, ensuring that every single transport vehicle is carrying the exact right chemical building block. With the amino acids securely attached to their transport vehicles and fueled up, the biological factory floor is fully prepped for production. This diagram outlines the first production stage. The 40S ribosomal subunit locks on to the mRNA's 5' cap. Carrying initiator tRNA, it slides down until encountering the start sequence, AAG. Then the massive 60S subunit drops down, clamping over the 40S subunit. Together, they form the complete 80S ribosome. This creates a fully enclosed decoding machine, perfectly aligned and ready to read the rest of the genetic tape. This animation shows the internal architecture of the ribosome at work. It features two primary operating slots, an occupied P site currently holding the growing protein chain, and an empty A site sitting right next to it. A new tRNA parrying the next amino acid settles into the empty A site, only if its anticodon provides a specific geometric match for the three-letter mRNA code below it. The moment it locks in, a component inside the ribosome acts like an industrial welder. An enzyme called peptidyltransferase instantly forms a peptide bond, transferring the entire growing chain onto the new arrival in the A site. Powered by GTP, the ribosome ratchets forward exactly 3 nucleotides. This ejects the empty tRNA, shifts the growing chain over, and leaves the A site empty, resetting the cycle. This process happens simultaneously along the same strand. These clusters, called polyribosomes, rapidly churn out identical copies of the protein chain back to back. This relentless, cyclic physical mechanism, reading, bonding, and ratcheting forward, is the literal engine driving cellular growth. The cycle only stops when the ribosome reaches a termination signal on the mRNA, specifically the codes UAA, UAG, or UGA. There is no tRNA equipped to read these stop codons. Instead, a release factor enters the ribosome. It triggers a chemical reaction called hydrolysis that severs the final bond, causing the entire ribosomal complex to dismantle into its separate subunits. The translation phase is complete. The cell is left with a newborn linear polypeptide chain. But a linear chain is useless until it takes on a physical shape. Specialized molecules called chaperones immediately begin folding the protein. During this process, proteins destined for the outside of the cell often reveal a specific chemical tag called a leader sequence. Looking at this diagram, a signal recognition particle grabs that leader sequence mid-production, docking the working ribosome at the endoplasmic reticulum membrane. As translation continues, the new protein is threaded through a membrane pore, securing it inside the ER to be modified and shipped. A highly regulated logistics network governs these movements, ensuring that manufactured proteins reach their specific intracellular or extracellular destinations. Because this process is so vital, it is a prime target for biological warfare. Lethal toxins like ricin and diphtheria kill by specifically jamming eukaryotic elongation factors. They instantly freeze the cell's ability to produce new proteins, leading to rapid cell death. Modern medicine exploits this exact same vulnerability. Antibiotics like erythromycin are engineered to intentionally jam bacterial ribosomes, which operate using slightly different subunits known as 30S and 50S. These drugs cure infections by halting bacterial protein syntheses, and they are safe for us because they physically cannot fit into our own 40S and 60S eukaryotic machinery. Understanding the molecular choreography of translation reveals the mechanics of human life and provides the exact blueprints we need to defend it.